Thanks for having me here. It's a pleasure. Good afternoon, everyone. I'm going to talk about something today that has been with us for a couple of 100 million years that we have been on this planet, and that's your gut microbiome. So over the past 10, 12 years, because of advent of gene sequencing, modern gene sequencing, faster, cheaper, better, largely by Illumina and others, we have figured out that we are actually symbiotes. Okay, we cannot live without our gut microbiome, and within the first 3 years of any baby born, if you don't have a functional gut microbiome, you will not develop a competent immune system, and it correlates with all kinds of chronic diseases, atherosclerosis, heart disease, diabetes, inflammatory diseases, including more and more CNS disorders, mood disorders, Parkinson, etc. So what's going on? Okay, well, you should view gut microbiome as your 11th organ system. Just like your other organs, if it goes out of whack, you're going to have problems. Okay, so the question is that we know it goes out of whack, largely because of our, of course, genetic basis in each individual, but largely because of our lifestyle, our Western diet and lack of exercise and so forth, and also aging. Okay, that's really happens to all of us, and it works just on every organ just like that. So what we were interested is look at it at the system level and figure out that since we know that if the gut microbiome, okay, dysfunction, it promotes various disease onset and progression, the opposite question we ask, is it true? If I can remodel the gut microbiome that is dysfunctional into a functional state, would it, prevent disease development? That's a good question, except there was no approach to it, and I will tell you what it is. Depending on our individuality, your gut microbiome state, based on this cartoon, may be here, mine may be here, and others over here. But collectively, if we have a healthy phenotype, and we fall into that zone. Now, a dysfunctional gut microbiome should be in a distinct state somewhere in here, and that, let's say, take my word for it, that would lead to disease. Now, the key here is, can I take a dysfunctional gut microbiome? And our gut microbiome has about 5 to 1,000 different species in it. And the genetic diversity of our gut microbiome is 100 times greater than our genetic diversity. So we are basically genetically about 1 or 2% human walking around. Okay? As far as the number of cells in our body, we're only 10% human. Okay? That's bacteria is small, you don't see it, but they are more than us, okay, by 10 to 1. So the question is, can I move it over here? Now, if I do this, the question is, does this give you a healthy phenotype? Does it prevent development of a disease? And how are you going to do this even? Okay, so none of this was known. And the question that you need to first address is, what is it that you call healthy or functional gut microbiome? And what is it, a dysfunctional gut microbiome? So we picked a small problem, heart disease, atherosclerosis, world #1 disease that in the developing world. And there's good animal models for it. Most of the statins, cholesterol-reducing drugs have gone through this model. This is a LDR receptor knockout mice. They're perfectly happy if you give them chow diet, which is a low-fat diet most mice eat, and they have healthy phenotype. But if you give a Western diet, which is what we eat, okay? But don't worry, don't feel any guilt about what you're eating. So you just give $50 million to Pete, he will have a pill for you in 5 years that will solve the problem. That's the bottom line. So if you give it a Western diet, they will have high cholesterol, plasma cholesterol, and over a period of 10 to 12 weeks, these are length of the studies, they develop plaques in their arteries. So the question we asked is that we know diet okay, influences your gut microbiome. If you change your diet within 2 weeks, your gut microbiome is remodeled. This is just a selective pressure that you're applying. But the question is, if this dysfunctional, if this remodeled gut microbiome is causative in the onset of this disease, then if somehow I can remodel this into here while still the animal is on the Western diet, then you can conclusively say for the first time that you can actually do this, okay, and it works. So it's simple. We put the animals on this diet and that diet. Within 2 weeks, we sequence the gut microbiome. And yes, they are very different from each other, completely remodeled. So we take this as a dysfunctional gut microbiome as a hypothesis. And then we want to do something to this to make it look like that, remodel them into that. So how do you do that? It's based on the things that I'm not going to discuss too much. It's cyclic D-alpha peptides that self-assemble into open-ended hollow tubular structures. And what we have found over the years is that they like to interact with membranes if you design them correctly. And some of these cause bacteria to grow slower, okay? Highly selectively, so it can find a sequence that interacts with this group of bacteria, but not these. Some interact over here, but not the others, and so forth. So this is a very unique class of molecules that you can modulate the growth potential of other bacteria. So the idea is, if I can modulate, slow down some bacteria that have been now more populated in the Western diet, keep them at bay, then the other bacteria can grow and rebalance the whole gut microbiome. Well, I wouldn't tell you if that didn't work, but we had to figure out how to do this study. So the way we do this, we take the cecum, which has part of the intestine that has large amount of gut microbiome, and we suture it, put it in an anaerobic chamber, open it, and use that content to inoculate every well in the 96 well. So every well has the entire gut microbiome as a system. We spend a year to figure out how we can grow them overnight without losing that diversity. And then to each well, now we add a different compound. We let them grow overnight, and then we sequence each one of these wells, just like we sequence me, you, and so forth. So we sequence each one and find out how it changed the gut microbiome at the system level. And then you do bioinformatics and so forth, and you find out what each peptide does to everything, all the species at the same time, find out which peptide brought the gut microbiome in the direction of the healthy, what we call the healthy state, the chow state, et cetera, et cetera. There's a lot of things behind this. And then we put them in animals, okay? So we select the top compounds that do it. So the animals are born and raised for about age 10 weeks on a normal mouse diet. At age 10, you either continue it on a chow diet, or you put them on a Western diet, or you put them on Western diet plus these compounds. Now, these compounds are not digestible at all, and they stay only in the gut. They are non-absorbable, et cetera. And then we study what happens to the animal. So I'm going to go through the slides very quickly. Within 2 weeks, you get 50% reduction in total plasma levels. This is amazingly high. All the bad guys go down, good guys go up. And basically, after the end of this 10 weeks, 12 weeks period, you hardly have any plaques in the artery beyond the age-related what you have in your control. Okay? So this is the maximum that can be seen in these types of animals. So it's extremely effective. How does it work? 68,000 genes that we can annotate in the gut microbiome is now changed. Okay? So the transcriptome is completely remodeled. We can do pathway analysis and find out and prove what sort of pathways and signaling molecules are changing that comes from the gut microbiome. In response to that changes in the gut microbiome, about 70,000 genes in the liver and intestine have been upregulated or downregulated, and we can do pathway analysis and find out how did you get rid of the cholesterol, and basically the remodeling of gut microbiome creates changes in the bile acid, and a couple of particular bile acid metabolites which are antagonists of its FXR pathway are activated, so your CPA71 keeps metabolizing your cholesterol. All the barrier functions are restored, your villi looks good, various receptors, 35 different chemokines and so forth, cytokines we have studied. Most inflammatory markers plummet to the basal level. Tregs are upregulated, and so forth and so on. So with that, I'll close and say that feel free to eat what you want. Just you have to hang on for 5 years, and Pete will take care of the rest of it. Thank you.